VC Chemistry, Unit 4, Enzymes, the highly functional dedicates. Okay. So enzymes are a biological version of catalysts. Like all other inorganic catalysts, they're only needed in relatively small amounts. They are not used up or changed at the end of the reaction. They do not alter a reaction's equilibrium position. Oh, do you still remember that? What's equilibrium position? It's the extent to which the reaction happens, which means that enzymes, just like all the other catalysts, they do not alter the conversion rate, so how much reactants were converted to products. That's the equilibrium position. But what they do is they provide an alternative reaction pathway that lowers the activation energy for a reaction. And therefore, they increase the speed of reaction, right? The rate of reaction. Remember, this is an energy profile diagram. We're using enzyme. When enzyme is catalyzed, then the activation energy is much smaller. Therefore, within the same period of time, you have a larger proportion of successful collisions. Okay, so enzyme is just a biological catalyst. But enzymes work slightly differently from other inorganic catalysts. So for example, one inorganic catalyst can catalyze many reactions. For example, if we use nickel to catalyze addition reaction of hydrogen, so however you change the alkenes, nickel will always work. However, one enzyme can only catalyze one particular reaction. Okay, so the following is a list of enzymes for 3 different types of food molecules. So for example, if you eat meat or seafood, you uptake a lot of protein. You need a particular enzyme called protease to break down the protein. And the fat part of meat and seafood needs to be broken down by lipase. So it needs to be catalyzed by an enzyme called lipase. So you can see that basically enzymes are named with a ASE at the end, and whatever that's before it is what it catalyzes. Okay, so protease catalyzes protein breaking down, and the lipase helps breaking down fats, and also amylase breaks down basically amylose and amylopectin sometimes. Okay, so sugar degrading enzyme amylase. There are a few terminologies we must be very, very familiar with to be able to describe what actually happens in this chapter. So first of all, substrate, that is a reactant. So this small blue chunk of cake is a reactant. And enzyme, this orange bit, is the catalyst. Normally, enzymes, they are a protein themselves. Okay, so where the substrate actually meets the enzyme, actually hugs the enzyme, that is where the interaction between substrate and protein occurs. And we call that the active site. So when it fits in there, and that's on this side, that's how the reaction gets catalyzed. So the interaction between substrate and enzyme is highly specific. It depends on their overall 3D structure. And since enzymes are proteins, so the 3D structure of enzymes are dictated by their tertiary structure or quaternary structure. Okay. So in VCE, what we need to know is there are 2 models describing enzyme action. The first one is the lock and key model. So basically, the substrate is the key, the enzyme is the lock. The key has to fit in the lock, otherwise it wouldn't work. So the shape of the substrate must match the shape of the active site, otherwise the key doesn't fit the lock. And the enzyme is specific for a particular substrate, so binding of a different molecule doesn't work either. Okay? So it's highly specificity. Okay? Another model is called the induced fit model. From my perspective, actually it's just a lock and key model with a little modification to suit what actually happens in the reality. So what actually happens in the reality is that the substrate doesn't really at all times fit 100% onto the enzyme, right? So what actually happens is that the enzyme, the presence of the substrate actually causes the enzyme to change the shape of the active site slightly to fit the substrate molecule. Okay, so the substrate still have to fit about like 90% or 95% onto the enzyme. It's not 100% exactly sort of like a perfect fit. There's always like the enzyme itself has to do a little bit of modification, changing the shape of its active site slightly to be able to fit or give a perfect hug to the substrate. But both models, whether it's lock and key model or induced fit model, they both demonstrate the specificity of enzyme action. So another perspective that shows the specificity of enzyme action comes down to the molecules that are chiral molecules. So we talk about that some molecules are chiral, and they have a chiral carbon, or at least one chiral carbon, in its entire molecule. So for example, this carbon that's connected to 4 different groups, that's the chiral carbon, and these 2 are optical isomers. So they are enantiomers of each other. So they differ in the way of binding to your taste bud, because we know that whether it's induced fit model or the lock and key model, we all very much focused on the shape of the substrate and the shape of the active site of that particular protein, right? So the shape of a molecule is really important in biological reactions, right? In the biological functions. So for example, these 2 molecules, they actually really differ by how that carbon is connected to the 4 different groups, in the order of the 4 different groups. But one of this molecule on the left, the molecule on the left can be bonded to your taste bud. As a result, you will feel sweet. You actually taste sweet. Okay? But the right molecule, because of the different shape of this entire molecule, it doesn't bond to the receptors of your taste bud. And therefore, it doesn't have a taste, because your taste bud doesn't receive that molecule. It's biologically inert. Okay? So enzymes are mostly proteins, and proteins are made up of amino acids. All amino acids, except for glycine in your body, are chiral, because all amino acids have a chiral carbon except for glycine. Okay? So basically, enzymes are chiral. Okay? Many substrates have more than one chiral center too. Therefore, enantiomers differ from each other in the way they interact with other chiral molecules. As a result, due to that, enzymes are very specific. Enzymes themselves are chiral, so they can only accept the other chiral molecules that actually fit onto their active site. If the chiral molecule changes to its own enantiomers, then it wouldn't fit onto the active site, and therefore it's biologically basically blind. It's inert. Okay? So due to specificity, often only one of the enantiomers is biologically active. So this kind of specificity is quite rare in chemical reactions. But in biochem, when biology comes into play, enantiomers actually makes a big difference. A drug with one enantiomer might work effectively. With the other enantiomer, it might actually worsen your symptom. Okay? So biological reactions are very specific. You have to have the correct enantiomers, because the shape of that molecule and of the active site of the enzymes are extremely important when it comes to whether it fits or not.